I'm Dimitrios Boumpas. I'm professor of medicine at the National Kapodistrian University of Athens, and my laboratory work is at the Foundation for Biomedical Research at the Academy of Athens in Greece. The SYSCID project is a big project. The role of my team is providing samples and input on a disease which is called systemic lupus erythematosus. This is the prototypic, as they call it, systemic autoimmune disease. Autoimmune diseases, they either affect a single organ or multiple organs at the same time. Lupus affects multiple organs at the same time, kidneys, brain, blood. That's why it's called systemic autoimmune disease. And the SYSCID consortium felt that they should have another autoimmune disease which is more systemic. It's not like rheumatoid arthritis, which predominantly affects a single organ, or the inflammatory bowel disease where the gut is affected, but they would like to have another autoimmune disease that affects multiple organs at the same time. In this project, we have collected samples from a group of over 300 patients with systemic lupus erythematosus. This is a disease of predominantly young women, because there are 7 affected women for 1 man. So it's a disease of young women. We have over 300 samples at the beginning of the disease, and when the disease is under therapy, and when the disease is quite down. The more important findings are the following. First, clinically, lupus is very heterogeneous. One patient doesn't look like the other. This clinical heterogeneity, we did find that at the molecular level. Through the analysis that we performed through the SYSCID, we have developed molecular signatures for at least 5 subgroups of the disease. So what we call clinical lupus consists of subgroups of groups, smaller groups, 5 different types of disease with a different molecular signature, with different abnormalities, which can be addressed more specifically. So that's the molecular heterogeneity parallels the clinical heterogeneity. That's a major finding. The second finding is that you could potentially use these molecular signatures to diagnose the disease earlier and also to guide your therapy, because different drugs affect these particular abnormalities that we detected. One of the important features of autoimmune disease like lupus, the same applies to rheumatoid arthritis, that the disease starts early before you see the clinical signs of the disease. So you have what we call a preclinical phase. And these molecular abnormalities that we discovered, they open the possibility that you could establish the diagnosis earlier. Early diagnosis means putting the fire off before damage occurs. That means better results and better rates of response in the patients. So early diagnosis and more specific diagnosis is an important potential application of this, because these diseases, they can be very difficult to diagnose. They can look like other diseases. So if you have a molecular signature, it helps the clinician to confirm the clinical suspicion. So early diagnosis and early treatment is an important part of this. The second is, these are lifelong diseases, and you need better treatments who are safe, but also they are effective. Finding out the right drug for the right patient in a lifelong disease, that means better results for the patient and less toxicity. And bear in mind that these diseases are diseases of young women, and if you use too much of certain drugs like cortisone, that can have side effects. So that helps you to tailor your treatment just to exactly to how much the patient and the right type of treatment will need. These are the 2 important. So better treatments, and the other thing, individualized treatment. Certain types of patients need a different type of treatment, and having a marker before you start treatment to pick these patients will be very helpful in the clinic. The findings of research, they have to be translated to the clinics. In order to do that, first we have to validate, to confirm your findings. What we're doing is was picking up another group of patients from the University of Crete with lupus patients to confirm that our findings are valid for them too. Once you establish that, then you can develop a test, a molecular test, that allows you the early diagnosis and the individualized treatment. And this can be done relatively easy now because the biotechnology is there. And once you establish this, then you have to confirm it again. It's a long process, but it can be done faster nowadays. The SYSCID tries to create a map of the diseases. For example, if you like to conquer a country, you need to have a map to know what it's about, where are the major cities, where are the rivers, the mountains. So this is the SYSCID. That was the ambition of the SYSCID. It was amazing to see that through a variety of techniques and variety of approaches, molecular, cellular, immunology, molecular biology, the SYSCID came up with a detailed map of the disease. So we know the pathways that we need to follow to reach an important target in the disease or an important organ that is being damaged. So it was fascinating. Initially, it did sound like science fiction, but it's very rewarding, I'm sure you'll agree with that, to see that it can be done. And after 4 years, we're very close to have detailed map of lupus in the case of the disease that we're interested. That's the first thing. The second thing is that for a clinician, it's very important to interact with the basic scientists and to share with them the clinical problems and also learn from them how could somebody approach them in the lab. This cross-fertilization between basic and clinical people was an important experience.